Everyone. Sorry I couldn't be there in person, but I'm very happy and honored that Andrew and Corey and the rest of the organizing committee thought you might like to hear about our Emerald Ash Borer Biological Control Program. So enjoy the presentation, and my contact information and how to get a hold of me if you have any questions are available at the end of the recording. So sit back, relax, and enjoy. So I'm going to be talking about emerald ash borer biological control in Canada and some of the next steps in our program. So my name is Chris McQuarrie. I'm a research scientist at the Great Lakes Forestry Centre in Sault Ste. Marie, Ontario, and that's my email address. So this is before I do talk about the biological control program, I want to acknowledge both the partner agencies and the businesses and the groups that have contributed to the program over the years. And this is just a selection of their sort of logos. And actually, this probably needs to be updated a little bit because I've noticed I don't have from some of our maritime partners on here. But there's been lots of folks involved in the program over the years, and very thankful. The team is also, we're also a big team of folks. It's not just myself. Over the years, we've had a large number of individuals participate in the program. So across the top row, that's our Sault Ste. Marie group. And then in the bottom row, we've got our insect production and quarantine laboratories team who grow the parasitoids and EAB. And then we've got our Quebec group. And then, of course, I think you might recognize those folks from the Maritimes. But because we've been doing this going back to just about 12, 13 years now, some of these folks have actually retired. So Barry Lyons started the program. Krista Ryle picked it up. Over the years, we had Gene Jones, who is our this is Gene right here. Gene, who is our technician extraordinaire, who ran the program for a number of years. Tim Ladd, who was our senior ring technician, and then of course John Sweeney, who along with Corey led the program out in the Maritimes for a number of years. Although for the number of emails I get from John, even these days, I think perhaps I need a question mark next to his retired status. Although I think he actually is retired, and we wish John all the best. So Andrew and Corey asked me to talk a bit about EAB biocontrol Canada and the next steps. And so what I'd like to do first is talk a bit about what we have been doing for the past 12 years and then give an update on what we know about how the insects are establishing and dispersing, and then close with a bit on what we want to do next. So for the next few slides, I'm going to show some maps of where we've released insects in Canada since the program began in 2012. And so this map shows the release locations of the most recent arrival, which is Spatheus gallinae, which we only began releasing in about 2017. And Spatheus is one of the two larval parasitoids of EAB that attacks the insects under the bark. And in all these maps I'm going to show, there's a dot for each release site, and it's colored by the year we first released insects there. So every time we do a release, there's 2 years of releases that happen. And so the first time we did releases, that's the dots. And what you might notice is there's nothing for 2020 in any of these maps. And I think the reasons for that are probably pretty obvious, and so I won't dwell on them. So this is just a zoom in to show the release locations in the Maritimes. Then these have all been coordinated by Corey and his collaborators over the past couple of years, and with the most recent releases being done in Cole Harbour and Moncton this year. So that's those kind of pale yellow dots on the map. And this is just a picture of Spatheus. You can see the ovipositor there that uses to insert its eggs underneath the bark of the tree and into the larvae of emerald ash borer as they develop. So this is the release map for Ubius agrilli, which is the egg parasitoid of emerald ash borer. And just for a sense of scale, in this picture here, we see this is an adult Ubius, and this is an emerald ash borer egg, right? So these are super tiny insects, and they're very hard to see, but they attack the eggs of emerald ash borer. So we've been doing releases of Ubius since about 2015, and you might notice that there's a few more sites in southeastern Ontario, kind of that Windsor, Guelph area, and that's sites where we didn't release Spatheus. And that's just because by the time that Spatheus was approved for release in Canada, there weren't any suitable release sites available down in that part of southwestern Ontario. Just because the ash populations had gone a little bit too far, had degraded a little bit too bit, and we didn't think there was places that could sustain releases. And so that's why we have Ubius released in a number more places than we do Spatheus. And here again is just that blow-up map showing releases in the Maritimes. And what you'll notice is that all the sites have received both parasitoids. And so what's interesting here is that the Maritimes have also received all Canadian-grown parasitoids of this species. So these are all been produced at our lab in Sault Ste. Marie. So these are all homegrown insects. For the first couple of years of the program, we got all of our insects from our partners in the United States at the Rearing Lab in Brighton, Michigan. And then about 5, 6, 7 years ago, we decided that we wanted to have some of our own homegrown populations, and so we started colonies, and we maintained colonies of Tetrastichus, which we'll talk about in a second, and Ubius agrilli at our lab in Sault Ste. Marie. And since about 2017 or 2018, we've been supplying releases. We've been supplying these insects for releases in Canada, and since that time, all of our Tetrastichus and all of our Ubius have come from our Canadian populations. Just sort of now here's a map showing where we've been releasing Tetrastichus. So in Tetrastichus planopenisi, it's a larval parasitoid like Spatheus gallinae. Uses that ovipositor to attack the larvae of emerald ash borer as they're feeding underneath the bark of the tree, and then the eggs develop on those larvae, killing it, and then you get more Tetrastichus emerging from those parasitized EAB larvae. And it's by far the most widely released and it's the largest number of releases for emerald ash borer biological control in North America has been Tetrastichus. It's been released all over the United States, and as you can see from this map, a number of places in Canada. And this was our first bug that we started releasing in 2013. So it's 10 years of releases of the insects in Canada. And what you'll notice when we show the map for the Maritimes, that all of these sites have received all 3 species. And this reflects the nature of the program since about 2017, where all of our new release sites have received all 3 species. And so the purpose of this is to target different stages of the EAB population and also host feeding in different tree sizes. So we have insects that are attacking both the larval stage and the egg stage. And then Tetrastichus, if you look, you can sort of see this very kind of short ovipositor right here, the part that it uses to lay eggs. It doesn't do real well attacking insects in kind of larger diameter trees with thicker bark. And so what Spatheus does, because it has a bit longer ovipositor. So what you do by having the combination of larval parasitoids and with an egg parasitoid is you're attacking all of those different stages of the emerald ash borer population, and hopefully beginning to have some of that level of control of the population, and reducing the impact of the insect in those regenerating ash. And so what I also hope you'll notice is that basically our pattern of releases across Canada has generally followed the expanding range of emerald ash borer. So the insect was established was first found in sort of Windsor and Detroit in 2002, and that's far southwestern Ontario. And as the insect has moved further east, we've also been making releases where there have been large populations of both EAB and ash that can sustain those populations of parasitoids until they can establish and begin reproducing. And so the goal of the program has never been to protect those large diameter ash trees that were present before EAB came along. The goal has been trying to establish these parasitoids so that regenerating ash on the landscape can be somewhat protected by EAB and can reduce the impact of those populations by the action of the parasitoids. So that's kind of been the overall goal of the biological control program in general. And the goal of our program in Canada has been to see, like, can we actually establish these parasitoids in Canada under our weather conditions and our forest conditions. And so, like I said, the goal of the program was to determine if these parasitoids could just successfully establish in Canada, and then later on we wanted to see, are they dispersing and have they had an effect on our EAB populations. So the question about where have the EAB populations been establishing is relatively simple to do. So since about 2015, what we've been doing is doing assessments of establishment. And what this involves doing is a couple years after the last release of the site. So remember I said we do two years of releases. We'll go back in and we will cut a couple of small ash trees, infested ash trees, bring them back to the lab, put them in some cages, basically see what comes out, see if we get our Tetrastichus or Ubius emerging. And we've been doing that since 2015. And from that, what we've determined for the most part is that most of our sites in Ontario and Quebec have seen the establishment of at least Tetrastichus, and in some cases both Tetrastichus and Ubius. There's a couple sites here in Ontario that we haven't had successful evidence of establishment for whatever, for various reasons. But for the most part, almost all these sites have had parasitoids established. We've had less success in determining the establishment of Spathius, and that's kind of for two reasons. We've made fewer releases, there's fewer sites, and some of those are still too young to be assessed, which includes most of our sites in the Maritimes. So, like I say, our first releases were done up at Siagas there in northwestern New Brunswick there just in 2019, and Corey will be going out to establish to maybe get the samples to determine that this winter. So we're just kind of at the stage on these far eastern sites being able to determine if they're established. And in the case of Spathius, there's a couple of other techniques that we'd like to try, which I'll talk a bit later in the presentation, to see if we can are we getting establishment of these populations at our release sites. But for the most part, that question is, have they been able to establish? Yeah, we think they've been able to establish in Canada. And then our next question is, are they dispersing? Like it's one thing that the insects are, establishing it in a population, but if they're in a place, but if they're not moving, they're not likely to have other impacts on EAB in other places. And so that's been one of the research questions that our lab's been looking at for a couple of years. And so that's basically examining dispersal. And so I chose this particular photo because it illustrates two of the tools that we've been using to determine how the parasitoids have been dispersing. So this is Megan Gray, who's the technician in our lab. And this here is the yellow pan trap. Pretty simple. It's just a shelf bracket that you attach to the tree, put this little soapy solution in, and then insects are attracted to the yellow, fall in, and then you come and you drain that off. And you, through a paint filter, and you examine the insects that you've caught. This other tool down here is what we call a sentinel bolt, and this is a technique that was developed in the United States by Jean Dewan and Claire Rutledge. And I'll talk a bit about how these work in a bit, but it's another newer tool to assess how emerald ash, how the parasitoid populations, and specifically the larval parasitoids, are establishing in a site. So beginning in 2019, we established a network of pan traps. So there's yellow pan traps. Here's another picture here. Around 7 of the release sites in southern Ontario, and then we monitored them over the summer for the presence of both Tet and Ubius. So what we did is we had this network of traps, and so the initial release site was at the center, and then we put pan traps at 1, 10, and 20 kilometers along either a northwest axis or an east-west axis around these 7 sites that had been established between 2014 and 2016. So these are sites that never got Spathius, so these are Tet and Ubius sites only. And then we monitored over the summer. So Megan and Danny, one of our summer students, basically spent the summer driving between all these sites a couple times and collecting all these pan traps. And then Megan spent most of the pandemic lockdown going through these pan traps at her house, trying to figure out what specimens we had caught. And then what we did in 2021 is we wanted to use these pan traps again, and we did a kind of a fine scale survey of the Algoma region in Ontario. So our lab, this is northern Michigan, this is northwestern Ontario, this is where Sault Ste. Marie is, so that's where our lab is. And so we established a fine scale survey of a number of sites in the Algoma region, and established looking at sampling away from our release at the very tip of St. Joseph Island. So this is a Parks Canada site where we've done releases. And we were looking to see how far they were spreading through this region, because this is basically our only release site within this part of Canada. There's an older one in Michigan just over to the south, but we were sort of examining dispersal from just this site. And so what I'm showing here is a map of those 2 release survey locations. So on this side, we've got our 2019 surveys and all of our spots in southwestern Ontario. And then over here on this side, I'm showing the Algoma region and all of our 2021 release sites. And then everything that's in black are sites where we didn't trap the parasitoid. Any parasitoids, and all the sites in red are sites where we caught at least one Tetrasticus. And so what the takeaway from this is that we've had reasonably good dispersal from a number of these sites. So we caught insects fairly far out from a number of these release sites. A couple of these release sites are like over lakes, so obviously we didn't catch anything there. But we have pretty good evidence of dispersal in both locations. And so what we did next with these data was try to compute, we computed the distance to the closest release site from each place where we caught parasitoids. And then we computed the time since release. So basically, what's the minimum amount of time it would take in a parasitoid population to disperse to that location. And then when we just fit a very simple regression to those data, we can calculate a dispersal rate, and we calculated a minimum dispersal rate of about 2.5 to 3.5 kilometers per year. And that's consistent with field observations from the United States and from flight mill studies. And so flight mill is just where you take a little insect and you attach it to kind of a treadmill and it flies around in a circle, and you can estimate how far it can fly over a period of time. And so all of our data were consistent with both field observations and lab observations. And so what this suggests is that the insects are dispersing kind of at the same rate or maybe slightly faster than in the U.S. But unfortunately, this pan trap method is really labor intensive. Takes a lot of time to go through all these samples. Takes a lot of time to process. They're kind of messy. There's a lot of downsides to working with these, and so we were looking to move away from them. And so what we wanted to try was this method that like say Claire Rutledge and John Dwan came up with and published about 2 years ago, and it's basically sentinel bolts. And so what these are they're just sticks of ash. So this is green ash that we harvest, and then in the lab we artificially infest them with emerald ash borer. It's the same technique that we use to rear parasitoids for the biocontrol program. And then we stick them in these cups, and this is just full of saturated rock wool insulation and sealed up with parafilm so that they don't dry out. And then we just hang them out in the field for 2 weeks and let during the flight time of the parasitoids just to see if we can get them to attack. And then everything comes back to the lab, and put them in a growth chamber, see if any adult parasitoids emerge, and then dissect them to see if there's any evidence of parasitism on the EAB larva. So what we did in 2023 was we tried these sentinel bolts at 5 sites over 4 different periods, 2-week periods in the summer. And we did these at duplicates of these. A lot of the sites we chose were duplicates of the ones we had sampled in 2021 because we wanted sites where we knew TET was present so that we could at least catch. We were confident we might catch something if the method worked. So this is our St. Joseph release site, and each of these stars is just where our release sites were. Again, here just is where our lab is. And so what we have here are counts of both the adult parasitoids on the right, and then we've got counts of the larval parasitoids on the left that we found when either we let the insects emerge or when we dissected the bolts. And then the 4 dates at the bottom are the sample periods when the bolts returned to the lab. And what we found evidence of at all dates is that, so we found evidence of parasitism at all the dates, but we got a bit better emergence of adults in our later samples, and we're really and then we did in our earlier samples. We did get lots of larval parasitism, and I was kind of wondering if we needed to let these ones cook in the growth chambers a little bit more to get some better emergence. But the take home from this is we're really excited about how well these work for catching TET. And we also caught 2 specimens that look like Spatheus, but we need to get those confirmed from our collaborators in the United States. But if this seems to work really well, and it was a lot less labor intensive than the yellow pan traps, and so going forward as one of the tools we can use to assess parasitism, this is a tool that we're really excited about being able to use. So I think we can say with some confidence that we've had pretty good evidence that the parasitoids are dispersing in Canada, and which sort of brings us to the next steps of the presentation and what's next. And if Andrew and Corey are watching the clock, I am just about to wrap up. So what's next? So we want to continue our release programs in Eastern Canada. So those sites that Corey established in 2023, we're going to get a second dose of insects this summer. We've also had conversations about adding us 1 or 2 additional sites in the Maritimes and perhaps 1 or 2 in Quebec as well. With our collaborator Vernick Martel there, who leads our program in Quebec. And we want to continue to use the sentinel bolt method to assess both dispersal and range, and this is something we're talking about for 2025. But the next big thing we want to do is examine the impact of these insects, and I'm curious to see if we can use these sentinel bolts for that. But what we have now really is this amazing set of release sites. They're all different ages, and what we can do is start to look at if we're having negative effects on the EAB population and if they're contributing to ash renewal in these sites. We kind of have this time series of sites that I'm really excited about being able to use to answer some of these questions. And finally, we want to be able to complete our program transition, and some folks may have heard me talked about this before. So the biological control program for EAB was initially begun as a research program, and the goal being to see could we get these to establish in Canada. And while it continues, the release portion is something we want to step back from and have collaborators take the lead on, both finding the sites and making the releases using insects that we provide. And so this would allow us to transition to some of the away from kind of the operational side of the biological control program, focusing on some of these research questions that we're interested in, and being able to make some predictions about how well it's going to work in the future, and what we can predict about ash renewal. So we'd started our program renewal a couple of years ago, and it kind of got put on pause by various things, and I'm hoping we can get back to completing either this fall or sometime early next year. So that's kind of the end of the talk. What I'd like to say thank you for listening, and I hope you found the presentation interesting. And if you have any questions, my email and my social media handles are right below. So thank you very much, and have a great day.